Hello. So Arcyria, and I want just immediately to make the connection between the my presentation and two previous ones, because in the case of Arcyria, it is also very difficult to find the type material, because many the core species are described several hundred years ago, and like Arcyria denudata, Arcyria panizia, and Arcyria obvulata, and so on, and Arcyria cinerea, of course. So the descriptions are very scarce, and the type material is either not found, or you cannot get any DNA from it suitable for sequences, at least now. So yeah, that would be another difficult taxonomic group and case. And first of all, what is Arcyria, and how it is understood? What is Arcyria, what is not? Actually, the understandings of the group changed throughout this history, and the taxonomical position of Arcyria was different. Some authors, like for example in Poulan et al., the Arcyria together with Pericana is one family, right? And in other systems, the Arcyria is just a genus of Trichiaceae. So, but usually when the people sees something bright spores, how we understand Arcyria it is or not. There are three main genera, and Arcyria is usually something with the stalk, with the cup, and with a capillicium that is like sponge. Trichia is, and also Arcyria could be really bright spores, like reddish, orange, and so on. Trichia is, and microscopically, the capillicium of Arcyria is the densely branched, dense net. Trichia is usually something with the capillicium as separate filaments. And Hemitrichia is a genus with the capillicium as usually a loose net of threads. And according to some authors, another distinction between Arcyria and others is the spirals on the capillicium. So it was thought that spirals are not typical for Arcyria; they are typical for Trichia and for Hemitrichia. Also, the birefringence of capillicium is not thought to be typical for Arcyria. Let us see how it will connect with the phylogeny and what the phylogeny will show us. The taxonomy of Arcyria is the topic of my PhD project. So I aim to build the backbone taxonomy and to put Arcyria in the backbone and then further to try to understand the morphological evolution of morphological traits for Arcyria. And then as the final stage to revise the morphospecies, because before the first two stages are done, there is little sense to revise morphospecies. So this is the geography of specimens that I studied. There are more than 550 specimens. Of course, not all of them were fresh enough to get sequences from them, but I managed to get sequences from of partial SSU, this barcode region, from more than 350 specimens, and elongation factor from 240. Yeah, and I also plan to get the third gene, cytochrome oxidase, but surprisingly, or not surprisingly, any pair of primers that I tried, and I tried many and designed many, they simply didn't work. I don't know why. I designed some new primers for SSU, they works, designed some for elongation factor, they more or less work, but no cytochrome oxidase. So we have what we have, two genes. And it should be said that the taxonomy of Trichiales was quite recently the good taxonomy, 3-gene taxonomy, was published by Ivan Garcia Cunhilos, whose work, which was the brilliant work. And he showed that he also analyzed some morphological traits and included some Arcyria to his phylogenetic tree. And he showed that Arcyria is in fact not a single branch, but 2 branches. And one of the branches of Arcyria branched together with some Hemitrichia clavata, Hemitrichia caliculata, and Trichia decipiens, which is also Hemitrichia now, as you probably already are aware of. But he was not focused like on Arcyria specifically, and he didn't make any taxonomical rearrangement specifically in Arcyria, because that should be done with the larger taxonomic on the larger taxonomic sample in Arcyria. So we decided to focus on the genus and try to make it with the larger sample. So what we had, of course, we confirmed the phylogeny done by Ivan Garcia Cunhilos. And we also got 2 main branches in Arcyria. So the genus is actually, it consists of the 1 branch, which includes all yellow species of Arcyria, Arcyria velata, fasciculata, pomiformis, and Phylogeny, and some of red sport species too, like Arcyria insignis and Erstedi. And the sister branch to this is a Hemitrichia, which includes the type species for the genus Hemitrichia clavata. That's what Ivan treated as the Arcyriacea sensulato, but of course it's, and as he mentioned, it's another branch. And we propose it to become another family. And since it contains the type species of Hemitrichia clavata, Hemitrichia, it might be a good candidate for a family Hemitrichiacea. Well, the other branch of Arcyria is Arcyriacea sensu stricto. And this branch includes 2 distinct, also, okay, 3 distinct subgroups. But on this picture, you see 2 of them. It contains such species, the red spores Arcyria, most of them are visual clade, and the other clade is gray Arcyria, which, yeah, we will also describe, I will also show in detail later. So 2 main subclades. And that is what we are not sure about that 100%, so it's not the final decision. It's just I proposed it for the discussion, because yeah, very well known that taxonomic changes should be very well thought of before they are made. But in our understanding, there are 2 families that include Arcyria-like taxa. It's Hemitrichiacea, which contains 2 genera, Hemitrichia and Heterotrichia. We propose to resurrect the genus name Heterotrichia masse for the Arcyrias with the yellow spores and some others. Yeah, it's not that easy. We don't have a clear morphological criterion to do that, but that's the reality. The genus the name Heterotrichia masse suggested for Heterotrichia Gabriella, which is the synonym of Arcyria Phylogeny. So that would be the type species. And Arcyriacea, in the changed sense, we propose to divide into the Arcyria sensu stricta, which would include the red Arcyrias. And the situation is a little bit more difficult with the gray Arcyrias, because this clade, the gray Arcyria, is not really the uniform one. It contains 2 subclades of gray Arcyrias, and we don't really know the relationships between 2 subclades. So there could be a decision to have one genus, Arcyria, because they are not red, they are gray or white. They can be white too, or possibly to have two genera. But we will figure that out in the near future. As for now, we will propose definitely have Arcyria, and this clade, another clade of gray Arcyria is still in Certocedes. So let's see the examples and let's see the morphology. So this is what we name a hemitrichiaceae. On the left, those are hemitrichias. Photos are not mine on the left, but the photos on the right are mine. And you see the those rows, not rows, columns. Those are species complexes. Those are not species, but species complexes. You see this Arcyria pomiformis. You can see 3, but there are actually 5 pomiformis like species. One of them is this Pausiaca that Karina and others sent from Australia, and there is 2 specimens from Martin also, the Pausiaca. And there is an obvelata complex, which includes obvelata, fasciculata, and some other taxa, some other obvelata like Arcyria. There is a Phylogeny versicolor complex, which includes Phylogeny, classical Phylogeny versicolor, and Australian Phylogeny like Arcyria. It's an interesting case because Phylogeny and versicolor are very distinct morphologically, but their barcode region is very similar. It's super conservative, and it differs only by like 3 or 4 species like signature. Yeah, exactly. Thank you. So interesting case. And there is the Insignis echtedi complex, which contains 2 species of Insignis with geographical preference, and the echtedi, which is a good species, one of not many. And these are examples of Scapelicia. The Capelicia are quite different, and I will show it also later. There is no clear structure that is like signature for the group in the Capelicium, so they can be really diverse. And there is the Arcyria sensu stricta, or the red clade of real Arcyrias. Those are also species complexes, and the row on the bottom, you see they are yellow, but they used to be red in fresh condition. It's just I don't have photos of fresh Arciodes incarnata, which is Arcyria congesta, or Arcyria occidentalis. But if somebody of citizen scientists has the photos of those species in the fresh condition, I have Minuta. I just have these photos of 2 species, then I would be really happy to get them, because I'm sure they are rosy or red, and I read it in descriptions, but in herbarium it just fades very quickly. So yeah, we have Denudata helvetica species complex, again with several species like taxa, Aphinis Mare complex, Stipata imperialis incarnata, and Minuta occidentalis congesta complex, which was Arciodes incarnata. So yeah, and all those need, of course, the separate revision. There's examples of Capelicia again, and you see all kinds of ornamentations, from the network to like spines and spirals and so on. And these are gray Arcyrias, one of the great clades. This is the Arcyria. Thank you. This is Arcyria. And you see that this clade includes several complexes, but and several species. Out of 14, at least 14 undescribed, it contains at least 14 undescribed taxa, but from described one, it includes Arcyria riparia, globosa, margina undulata, and Arcyria follicola, which was described as Colonema follicola, which this Colonema case was already shown by Ivan Garcia Conquillos. But it's what about the other gray Arcyrias? They are usually identified by everybody like Arcyria cinerea. But we don't know where this type specimen, what is the type specimen of Arcyria cinerea, and this description is so scarce. So we definitely cannot put any of these other undescribed species like taxa to match it with Arcyria type specimen of Arcyria cinerea. So this is the problem. This clade, this species have very extremely variable SSU region. So, and they are often situated on like long branches. So we will try to disentangle this in future. And yeah, but be ready that among Arcyria cinerea there will be many twin taxa, twin species. And those are examples of Capillicium again. You see this Riparia Capillicium, which is covered by spirals and spines, and spirals, spines arranged as spirals, and so on. And finally, there's the enigmatic clade of gray Arcyrias again. This is again Arcyria cinerea mostly, but also Arcyria afralpina on the top on the right, and the species that is not accepted currently, but I believe in this species, Arcyria carleta. It was described by Hertel in the middle of 20th century. This one is a right bottom one, brownish, creamy, and this Arcyria expands from the top, but retains the form of the sporangium on the bottom. If I find this green dot. Okay. Carleta. I think I should do this. This one. So, and Hertel specifies that it has kind of a peridium, not like real peridium, but something peridium-like structure from the spores that are collapsed, but like retain as the peridium in this species. And it's exactly, it's like a very stable characteristic. So we have about 10 specimens that are like that, and they form a good clade on the tree. So I believe in this Carleta species, definitely. But others are also like, this is Arcyria cinerea, I would say, and this also looks like Arcyria cinerea, and this is something more olivaceous and with other Capillicium. But that's what we have. And examples of Capillicium. Here you can see like usual cinerea type, but also this is the afralpina, and there is this olivaceous olivaceous species with the reticulate Capillicium. And the last clade that we discovered unexpectedly is the clade based on single specimen that was collected by citizen scientist Tyson Necklers in British Columbia in Canada. He collected something that nobody could identify even to the genus level, and including me. So he sent it to us, and after sequencing and morphological examination, it appears to be a new genus. It's a clade that is in Certicedes, so it sometimes groups together with Arcyriaceae sensu-stricta, and sometimes with Arcyriaceae and Trichiaceae. So it's a strange clade, but definitely it is something separate, and it deserves to be a genus, because the Capillicium attachment, the Capillicium itself is the very unique. It doesn't form dense network like in Arcyrias. It's like very extremely elastic, and it's coiled in the spirals all the way. And you see how it protrudes from the sporangia. And that is why we choose the genus name, generic name Spiromyxa from those characteristic spirals. And it's a capillation covered with this rings. Not very typical for Arcyria. It happens sometimes, but quite rarely. And this Spiromyxa slocanensis, the slocanensis comes from the Slocan Valley, which is the favorite collection spot of Tyson Ehlers, apparently. Yeah. So that's what the phylogeny shows. And what about morphology? When I thought about the characters that allow to distinguish in Trichiales and in Arcyria, I thought of two groups of characters. These characters usually are used to distinguish between Arcyria, so within the morphological genus Arcyria. So you see there how the Arcyria grows. Is it on grows straightly, or drooping, or forms the digitate structure or the compound fortification? Also, the ornamentation of the inner side of the cap is usually used in the morphological keys. Ornamentation of capillicium, and attachment of the capillicium to the cup. It could be easily blown away or attached firmly, as you remember from the keys. And spores. Those are Arcyria, spores in Arcyria are quite typical. Most of the species have this kind of spores with two types of warts, but sometimes some species have this or these types of warts, like Arcyria pseudoleucarpa, hemitrichia, which is Arcyria pseudoleucarpa. And this is a hemitrichia, the schematic hemitrichia spores, just to put on the tree. So there is another picture of the same tree. With every sequence here in Arcyria is representative for the cluster of sequences, for the species, let's say, species like taxon. And here I put the those morphological traits on the tree. Apparently, so yeah, apparently there is no clear tendencies. So you see that the capillicium could be in heterotrichia, could be either reticulate or the spiny, or like spiny reticulate or spiny tall, and the ornamentation of the cup could be quite different. And the mode of growth is either drooping or like erect or stacked. So, and let's look at other red Arcyria. It's the same. And what you see specifically is that the spiral ornamentation of capillicium has evolved several times. It's in unrelated clades. So you see those warts or spines or reticulate ornamentation on everything else. And also you see the drooping and the erect, the compound spore curves. You can see all the types of those structures on the tree. The same situation is with gray Arcyriacea. Several times you see spiral ornamentation, spines, and sometimes reticulum. So yeah, what the conclusion we can make from it? Those morphological characters will work only on lower taxonomic levels, on the species level, probably. But they do not really help on higher taxonomic levels. They do not help to distinguish between the genera or even like sections of the genera. So the other group of characters that we try to reconstruct, and similarly to the work that did Ivan Garcia, we reconstructed the morphological characters that could be mapped on the trachealis. We didn't reconstruct those that Ivan already did, but we tried to understand some other characteristics. And we discovered several interesting facts. For this reconstruction is the states chosen as for the reconstruction were sessile, stacked, or compound or colonial sporocarp. And you see those. This is the Arcyriaceae sensu stricto, and these are Hemitrichiaceae. And you see, and this is Trichiaceae. The stalk, which is in blue, appears many times in the phylogeny of Trichialis. It's in Metatrichia, it's in some Trichias, it's in Arcyriaceae, it's in Hemitrichia, and in Heterotrichia. And the tendency to grow colonially to form the compound sporocarps, compound fruiting bodies, also emerged several times. It's of course in Dictydietaliaceae, and it's emerged sometimes in Arcyriaceae. Of course, in Arcyria congesta and Arcyria accidentalis. Of course, this is somehow, well, there is no clear border. Should we treat the Arciodes incarnata as this separate sporocarps or compound fruiting body? But I speak here about tendencies, not about like clear borders. So we see that the tendency to colonialism is here, and it emerged several times. The other reconstruction was the arrangement of capillicium, which are divided into several types. This is Arcyria-like type, net attached to stalk, and also this is most of the Dianemataceae have this kind of capillicium attached to the peridium, and the separate filaments like in Trichia and Luxnet and Epson. And what you see that this dense net-like capillicium is reconstructed is as the ancestral state for the not only for the Arcyriaceae and Hemitrichiaceae, but maybe for the Trichiaceae too, for the part of the Trichiales that includes all these three groups, which is interesting thing and something to think about. And the last one, the last character are pigments. Of course, this tendency is obvious. There is red Arcyriaceae, there is gray Arcyriaceae, and there is yellowish Hemitrichiaceae. And this character probably is most of all characters consistent to the higher taxonomy of Trichiales. But it's there is nothing. There are always exceptions. For example, among the gray Arcyrias, there is one yellow or orange Colonema Arcyria follicola. So it's like partially consistent. And you see, Heterotrichia is quite also quite. And it's not a uniform group. It includes mostly yellow spored species, but some with red spores. So that's the reality. So we in future we plan to revise this clades of Arcyriaceae and do the species complexes revision separately for all those groups. But for now, these are the results that we have. And thank you for your attention. And thank you to everybody who shared their specimens. And yeah. the citizen scientists and of course the academic scientists from the herbaria that shared the specimens to us.
Thank you very much, Iryna. Your questions, dear friends?
So, as you said, morphological traits can use to distinguish samples at lowest taxonomical levels, but there are many twin species in at your presentation. So, if we can't use morphological traits to determine samples to genus, and it's difficult to determine to lower level, only way to distinguish species is genetic study, isn't it?
It is sometimes, and sometimes it's not. There are, if we are lucky, we have a good morphological species that can be distinguished by the one prominent trait or by the combination of other traits. But sometimes we have the twin species that we cannot morphologically distinguish, at least with those characters that we can detect. For example, the Licagala case is a very similar one, but Dmytro managed to find the character that actually can recognize, can make the difference. In Arcyria, I couldn't find this character that would allow to, to put the species in. Like, I'm not that lucky. I don't have like this peridium structures that the Licagala have. Maybe I will find, or maybe.
I have a question from Ivan Garcia-Confilos, and I will read it aloud. So he says, what an excellent work, Iryna. Congratulations for it. I have two questions. First, did you try to get longer 18S sequences, especially for the clade you name Heterotrichia?
Not yet. I didn't. Maybe we have some hopes for even longer sequences, but it's not done yet.
And his second question is, I understand that with your expanded dataset, morphological features did not distinguish the red Arcyria, e.g. Arcyria denudata, and the clade containing some of the yellow spored species plus some of the other reddish species, besides color for some species. So have you checked if there are some ecological differences you can trace? Thank you. So.
Yeah, this is a good question. That's what we. This is the most difficult part. The only character that somehow helps in distinguishing these two groups is pigmentation. So among those gray and red Arcyrias, there are, for now, there is no like really brightly yellow spored species. There is the Colonema follicola, which as far as I understand is like orange something species. But those most yellow spored are here. But this is the only character that distinguishes them. All others do not. And but the thing is the phylogeny does, and that is why we have to finally come to taxonomical conclusions, because this is what phylogeny says. We cannot ignore it.
Yeah, thanks for this wonderful, very interesting talk, which again, of course, shattered our ideas how we can easily distinguish species. The same pattern we see in Lamproderma, the same pattern we see in Physoralis. For example, many characters help to distinguish species, but will not help us with higher taxa. So a very practical question I would like to discuss later, of course, in the discussion is how we construct keys now with these characters. But my special question here, Arcyria pomiformis and Arcyria cinerea, which are two traditional species, Farr has long time ago tried to distinguish them, since they are sometimes very difficult to distinguish. Right now, according to your results, it would be super easy on behalf of molecular signatures. Is that true?
Yes, it is true. It is absolutely true. They even belong to different genera. And moreover, well, for me, at least for me, it has never been difficult to distinguish between pomiformis and cinerea. They are morphologically very distinct, especially when you look at the inner cup ornamentation. It is like they are different. Yeah, well, I'm not really like an ecologist, but the morphology is distinct.
When you've been studying, sometimes you read that the ornamentation of the capricium varies from the bottom near the colliculus and upwards. Are you looked into the variation, and have you been sort of consistent in where you're collecting the fragments of the capricium when studying the ornamentation?
Yeah, thank you for the question. It's a very common phenomenon in Arcyria, and you have it in many species, in cinerea, in like all the cinerea clades, in Phylogeny, in many other species. So the capulicium closer to the cup, and the inner capulicium is usually thicker and less prominently ornamented, and even sometimes smooth in comparatively to the outer part of the peridium. Well, it's how to put it? I believe it's not. I cannot prove it, but I believe that this is the character that is regulated ecologically, but it has some a variation. It has some within species. It's also for some species it is very distinct, and in other species it's not that distinct. So it's helpful. It's helpful. There are some. I will show you from those cinereas. In this clade, there are species that really have this distinct, always have this distinct difference between outer and inner capulicium. So yeah, it can be used as a helpful part of the species description.
Iryna, several questions from me. First, the shortest one. If I understand well, the spiral ornamentation of capricium helps the tube to do a hygroscopic moves. At least those species which have these spirals, like Trichia varia, may move their capricium. What can be a function of Arcyria types of ornamentation, like these rings, half rings, spines? Because they are too prominent to have no function.
Well, it has something to do with spore dissemination. Well, I think that Arcyria capulicium functions a little bit different from the trichia and from this kind of movement. So it expands like a spring, more like a sponge, and I'm not sure if any study has shown this kind of hygroscopic movements of capulicium in Arcyria. So it somehow helps spores to disperse.
You suppose it may help to do this initial jump?
Or maybe not. Maybe to keep spores some spores inside while the outer spores are disseminating, are leaving the sporangium, and some spores are kept away, and then those other are slowly, gradually disseminating.
Could it also help to avoid the fixation of the spore on the thread, detaching of the spore from the thread, because they all are formed all together, and the spines can prevent spore to attach when it is wet, to
They're
attach to
kind
the
of
capillicium.
hydrophobic, the
Mhm.
spores of Arcyria. So,
But spores are not highly ornamented. So maybe this hygrophobicity and the detaching function is mainly made by capillicium.
but they never attached to the capillicium. They're like
Yeah, because it is like this. If it would be smooth, what could prevent them to attach capillicium?
you mean
Yeah.
That's possible. That's possible to keep for some time, to keep them inside the sporangium, to allow to gradually be blown away by the wind. That's I believe firmly. Yeah.
Okay. Second question. We discussed this, but I want to share this discussion. When you can you show please a color tree of spore color? Here we clearly see that the white color, great white color, emerged only once, and the red color emerged at least twice, or even more, even more. What is your opinion about the yellow color? Because it is already present in Dianemataceae, in some Dictydentalia has a yellow spores. There is also yellow color in Hemitrichiaceae branch and in Trichiaceae branch. What do you think? Is it just a plesiomorphy? It's just a basic characteristic of the whole order or not?
And moreover, there are some brown branches, but brown, as we know, is the shade of yellow. It's also a yellow. So there should be always a caution that a color is just something that we see, that we perceive, but the color could be caused by different sorts of substances, of course, and those characters could be not homologic. But we have to start from somewhere. And yeah, this yellow pigments are reconstructed as the basal state here. And if we treat a brown as a yellow, they would probably be also here for the trichialis. And it can be, it looks really plausible that the yellow pigments are the ancestral state. And to prove that, we should analyze some representatives of from all this tree and show either show whether Arcyria flavins of Arcyria of yellow Arcyria are the same pigments as cause the pigmentation of Trichia and Hemitrichia and Dianema, and we will know.
Yeah. Okay. So chemistry may help.
Yeah, chemistry may.
Yeah. And the last question, the most discussable. Red true Arcyrias and two branches of white Arcyrias form a monophyletic clade in all your phylogenies, right? White and red, yeah. Albarcyrias and true Arcyrias. Yes. So what is the reason to separate this branch on 2 or 3 genera?
The reason, well, we don't have a clear criteria to separate genera, but if we will talk simply about the genetic distances, for example, so we will see that the genetic distances between the genera of Trichiaceae, at least those that were like more described or revised by, let's say, Ivan, and then those distances within Arcyriaceae are the same, comparable, or even greater than those within Trichiaceae. So, but these are only distances, and we have here a morphological evidence that would make it quite easy for everybody to distinguish between the genera. This is quite a good case. this is a bad case.
Any other questions? Thank you very much, Iryna.